Professor Lynn Morris from the National Institute of Communicable Diseases is joining me on Google Hangout. She's recently taken part in a study that has brought us one step closer to finding a vaccine for HIV. All right, Lynn, you recently did a study and you focused on a woman's body. She was from Kwazulu Natal, and the way her HIV infection responded to potent antibodies. Can you just elaborate on this and explain what took place in the study for us?
Right. Yes. So this is the study of a single woman, because the antibodies that she developed in response to her HIV infection were very potent and very broad. And what that means is that her antibodies, when we test them in the lab, were able to kill viruses from around the world, because HIV is a very variable virus, and it's a bit different in different parts of the world that they call different subtypes. But what her antibodies were able to do was to recognize something common on all of these different viruses. And the reason why that's interesting is because those are the sorts of antibodies that we want a vaccine to induce. So by studying her particular samples, and we had the good fortune she was a participant in a study, and she came and donated blood right from when she first got infected with HIV, and on a regular basis donated blood that were all stored in the freezer. So we were able to go back to the freezer and actually figure out exactly how her body made these antibodies.
Okay, and these are also known as broadly neutralizing antibodies. What percentage of people who are infected with HIV would you say have these types of antibodies?
That's a very good question, because it's only about 20% of people who have HIV make broadly neutralizing antibodies, and we don't understand that. We don't understand why some people make them and other people don't. But I think it's also important to just say that the people that make them, there's no clinical benefit to making them, because their disease progression is unaltered. In other words, it doesn't help them to control their virus any better. And the reason for that is because HIV is always a step ahead of the immune system. And so it's already escaped the antibodies. But what we do know is that if we could induce these kinds of antibodies by vaccination in an HIV negative person, that person would be able to fend off HIV, because the antibodies are there before the virus.
Now, Lynn, what were your findings once the study was complete?
So what we found is the antibodies from this particular woman were highly unusual. And they had what are called long CDRH3s. Now that's a very technical term, but what that means is they had very long arms or very long protrusions. And we know that this is a strategy that the human body uses to fight HIV, because these kinds of antibodies have been described before. And what these long arms allow the antibody to do is to penetrate the sugar coating that HIV covers itself in. So that's the defense that the HIV virus uses to fend off antibodies. But what these antibodies have done is they've grown a long arm, and this long arm basically can bypass that defense. Now, we have known that this is, that these kind of antibodies do develop to HIV. They were first described, in fact, in 2009. But what our study has shown, and this is because we had these stored samples from this woman right from the time that she got infected, we were able to figure out how the body actually made these long-armed antibodies. And so that is the key finding. And the way that the body did it, in fact, is that they selected B cells. So B cells are what makes antibodies that already had the long arms. So these do exist in our normal repertoire. And so these particular B cells were selected already with the pre-existing long arms, and that kind of gave this antibody a jump start, because all it needed to do then was just have a few additional changes, and it was then able to kill HIV.